हेलो फ्रेंड्स, टारगेट 90 प्लस सीरीज़ में आपका स्वागत है। आज हम चैप्टर नंबर 11 एनहांसमेंट ऑफ फूड प्रोडक्शन देखेंगे, जिसका वेटेज 4 to 6 मार्क्स है। लेट्स सी द क्वेश्चन आंसर्स। व्हाट इज प्लांट ब्रीडिंग? द मैनिपुलेशन इन द हेरिडिटी ऑफ क्रॉप एंड production of new superior varieties of existing plant is called breeding. What is biofortification? It is a method in which crops are bred for having higher levels of vitamins, minerals, and fats. Name two amino acids found in fortified maize variety: lysine and tryptophan. Name two acids produced by Aspergillus niger. It is citric acid and gluconic acid. Give full form of MOET, multiple ovulation embryo transfer technique. Name the organism present in idli, dhokla and dosa: Leuconostoc and Streptococcus. Name the organism producing lac. It is Tracharia lacca. Give the products of apiculture. Apiculture means beekeeping, honeybees, honey, wax, pollen, bee venom, bee glue and royal jelly. Name the best quality silk and inferior quality silk. The best quality silk is mulberry silk and inferior quality silk is tussar silk and eri silk. What is the main function of fermenter? The main function of fermenter is to provide controlled environment for growth of microorganism to obtain desired product. What are heterocysts? Heterocysts are colorless cells present in anabina that help in nitrogen fixation. In 2 to 3 mark questions, name the organism present in Roquefort cheese, Camembert cheese and Swiss cheese. In Roquefort cheese, Penicillium roqueforti. In Camembert cheese, Penicillium camemberti. Swiss cheese, Propionibacterium shermani. Give the examples of fortified plants and their contents. So, wheat Atlas 66, it has high protein content. Iron fortified rice has 5 times more iron. Carrot and spinach have more vitamin A and minerals. Bitter gourd and tomato have more vitamin C. Enlist types of poultry diseases: viral diseases, ranicate, bronchitis, and bird flu; bacterial diseases, cholera, typhoid, and TB; fungal diseases, aspergillosis and thrush; parasitic diseases, infection by lice, roundworm; protozoal diseases, coccidiosis. Give example of natural mutagen and chemical mutagen. Natural mutagens are high temperature, high concentration of CO2, X-rays and UV rays. Chemical mutagens are nitrous acid, EMS, ethyl methyl sulfonate, mustard gas and colchicine. Sketch and label Shibboleth tower fermenter. So this is the diagram which consists of fermenter and top layer alcohol, yeast and heat exchange pump. Sketch and label biogas plant. Biogas plant consists of digester, gas holder and pits. Explain the process of sewage water treatment before it can be discharged into natural water bodies. So there are 4 steps. Preliminary treatment, in which 2 steps are there: screening and grid chamber. Screening में floating suspended materials filter करते हैं, whereas in grid chamber it is made up of stone and pebbles and the coarse particles are settled down by gravity. In primary treatment, फिर प्रिलिमिनरी ट्रीटमेंट के बाद वो प्राइमरी ट्रीटमेंट में जाता है. वहाँ पे water is pumped into primary sedimentation tank in which organic and suspended matter is settled down. This is called as primary sludge. The secondary treatment, biological treatment, in which effluent is constantly mixed mechanically and air is pumped. The bacteria grow and form the flocks, and this decreases the BOD. Tertiary treatment, In this, effluent is passed to the settling tank where bacterial flocks are removed by sedimentation, and the sludge is called activated sludge. State the benefits of biofertilizer. Low cost and can be used by marginal farmers. It increases soil fertility. BGA as biofertilizer secrete growth promoting substances like organic acids, proteins, and vitamins. Azotobacter supply nitrogen and antibiotics in the soil. Biofertilizers increases physicochemical properties of soil. It also increases water holding capacity of the soil. It does not cause any pollution. Describe the structure of biogas plant. The most biogas plant model is KYIC and IARI. A biogas plant consists of digester and gas holder. Digester is made up of concrete, brick, cement, or steel. The cylindrical gas holder or gas tank is present above to collect the gases. The digester has side opening into which raw materials is fed. The digester is partly buried in the soil. स्टेट 3 बेनिफिट्स ऑफ यूजिंग बायोगैस. यहाँ पे मैंने बहुत सारे मेंशन किए हैं, आप कोई भी 3-4 लिख सकते हैं अकॉर्डिंग टू द मार्क्स. इट इज़ चीप, सेफ, रिन्यूएबल सोर्स ऑफ एनर्जी. इट कैन बी easily generated, stored and transported. It can be used for domestic lighting, cooking, street lighting as well as small scale industries. It burns with the blue flame and without smoke. It helps to improve sanitation of the surrounding. It is eco-friendly and does not cause pollution and imbalance to the environment. The sludge which is left over is used as biofertilizer. What are biocontrol agents? Mention biocontrol agents and their host. The natural method of eliminating and controlling insect pests and other diseases causing agent by using natural biological enemies is called biocontrol agents. Examples are given here. Fungi as herbicide. Phytophthora controls milkweeds in orchids. Alternaria control water hyacinth. Fusarium control most of the weeds. Trichoderma are free-living fungi that are found in the root system. Bacteria as insecticide and herbicide. Bacillus thuringiensis is used to get rid of butterfly and caterpillars. Pseudomonas and Xanthomonas attacks several weeds. Insects as herbicides. Tyrium moth control the weeds. Give the applications of tissue culture. The different applications of tissue culture include production of disease-free plants and plantlets, micropropagation, production of secondary metabolites, protoplast culture, culture of rare plants, somaclonal variations, production of stress-resistant plants, etc. What are biofertilizers? Explain them with example. Biofertilizers are commercial preparation of ready-to-use live bacteria or fungal formulations. Examples: nitrogen fixing biofertilizer or diazotropes. These organisms convert atmospheric nitrogen into nitrogenous compound like nitrates and nitrites. Symbiotic nitrogen fixing microbes, Rhizobium and Abina. Free-living nitrogen fixing bacteria, Azotobacter and Nostoc. Phosphate solubilizing biofertilizer. These bacteria solubilize insoluble phosphate from the rock. Example: Pseudomonas. Explain the process of biogas production. इसके 3 स्टेप्स हैं. पहला है हाइड्रोलिसिस, जिसमें रॉ मटेरियल ऑफ cattle dung is mixed with the water and slurry is made. It is fed into the digester. The bacteria like Clostridium, Pseudomonas hydrolyze carbohydrate into simple sugars, proteins and amino acids. Second step, acidogenesis, in which the anaerobic bacteria convert simple organic material into formic acid, acetic acid, H2O and CO2. In third step, methanogenesis, the anaerobic bacteria like Methanobacterium and Methanococcus convert acetic acid, formic acid into H2O, CO2 and methane. Give the advantages of single cell protein. These microbes can be easily genetically modified to vary their amino acid composition. They have high protein content. They can be grown on waste material. It serves as good source of vitamins, amino acid, minerals and crude fibers. The microbes have high rate of multiplication, therefore in short duration we can produce large quantity of biomass. Describe the methodology of tissue culture. So first point is cleaning of glassware and sterilization. In second point, selection and preparation of medium, MS medium. Next, sterilization of medium by autoclaving. Preparation of plant material, that is explant, by sterilization. Inoculation of the explant into the nutrient medium. Incubation of the inoculated explant. This forms the callus. Then organogenesis to initiate rooting and shooting. Hardening. The plantlets are transferred to polyethylene bag. And last, transfer to field. Write the chemical reactions to represent the methanogenesis. First reaction, hydrolysis, in which methane is formed. Name the microorganism used as SCP, single cell protein. Fungi, Aspergillus niger, Trichoderma viridae. Yeast, Saccharomyces cerevisiae, Candida utilis. Algae, Spirulina, Chlorella. Bacteria, Methylophilus and Bacillus. Name the microorganism used to produce organic acid. Citric acid, Aspergillus niger. Gluconic acid, Aspergillus niger. Fumaric acid, Rhizopus rhizus. Acetic acid, Acetobacter aceti. Name the vitamins with their microbial sources. Vitamin B2, Neurospora gossypi and Aromathecium ashby. Vitamin B12, Pseudomonas denitrificans. Vitamin C, Aspergillus niger. Name the antibiotics produced with their microbial source. Chloromycetin, Streptomyces venezuelae. Erythromycin, Streptomyces erythrus. Penicillium, Penicillium chrysogenum. Streptomycin, Streptomyces griseus. Griseofulvin, Penicillium griseofulvum. Bacitracin, Bacillus licheniformis. Oxytetracycline, Streptomyces aurifaciens. Name the enzyme produced with their microbial source. Enzymes कौन से बना रहे हैं? Invertase, Saccharomyces cerevisiae, Pectinase, Sclerotinia libertine, Aspergillus niger, Lipase, Candida lipolytica, Cellulase, Trichoderma conigi. Give the flowchart of sewage water treatment. So you can draw this flowchart. All the best.